So I'm honored to present Laura Pollock on the second keynote of the day. She's a professor in McGill, and we are honored to have her here today with us and listen to her perspective on a more ecological applications for biodiversity conservation and much more. So thank you very much, and please let's welcome Laura. Thanks, Kim.
So I promised I would talk a little bit about conservation, but after listening to the talks this morning, I just want to talk about model evaluation nonstop. But I won't. So I thought I would give you, I think a lot of you kind of know we're doing this for a reason. We're doing this in the end for conservation, maybe ecology too. But kind of what is it? Maybe sometimes. What is it that's kind of useful from what you're doing? And I feel like there's so many amazing, brilliant minds in this room that are focused on the things that I think we need to focus on right now, which is how to make these models. But there are so many applications for these models. And sometimes it's useful to think about the application when you're making the method. And so that's what I want to kind of try to convince you of today a little bit. So I'm just going to talk about a few things that we work on in the lab, that we work on with collaborators. And I'm not going to talk that much about 30 by 30 because I feel like we talk a lot about 30 by 30, but spatial planning, so systematic conservation planning, spatial planning, I think is one of the best applications for SDMs. And it's one of the most, it's kind of the only way we can do it. You can use IUCN range extent if you wanted to, but that's just really coarse. And SDMs do a really good job at this. And right now is the time to do this because we have the Global Biodiversity Framework. There's efforts in Europe to do this. So just a quiz to start you off after lunch. How much biodiversity was protected in 2017? What I mean by biodiversity is how much of the range, the spatial range of every species, by every species I mean terrestrial vertebrates, was protected in 2017.
Do you mean total or the new stuff?
On average. 10%. Average 0.1 globally in protection. 20? 2.
1.
Oh, that's really 1. Okay, you guys, it's way better than that. It was 10%. And so 10% is considered like very, very minimum viable amount that you would protect. That 10% is not distributed evenly. So most rare species are not even close to 10% in protection. So how much is protected now? Oh, I gave it to you. About 25%. Is that about right? It's about 25%. Why is that? Did we protect more species between 2017 and now?
Did we name more things protected?
We named more things. So we switched our definition of what is protected now. It used to be sort of categories 1 and 2, no take, like protection for the sake of protecting the animals and the everything else. And now it's switched to OECN. So it's a much broader. So that's kind of what we're working with, but at least it's set now. And so 25%, if you start to look into what some of these protected areas are, they're not really protected at all, but at least there's something. They're better than nothing. If you want to know more about this global results, talk to Isaac, who's right here. It happens to be there. So it's like top secret stuff, but you should talk to him about it. So the Global Biodiversity Framework, I just want to mention this because I think not enough scientists are paying attention to what's happening in this sort of policy space. And yes, it's a big, messy policy space, but there are very few scientists. If you go to any of these meetings, it's mostly policymakers. Sometimes it's NGOs, but very rarely do you have someone that actually could tell you any details about any of the data. And so these are goal A, goal B, goal C, goal D. The big ones for us are usually goal A. So goal A is kind of a biodiversity one, and then goal B is more of like the ecosystem services perspectives. But within each of those, there's a whole bunch of other targets and subdivisions that I'll talk about a little bit. But the reason I think that SDMs are useful for these kind of large scale efforts is because they want to know things like how much biodiversity do we have? How much might we lose? Is it increasing? Is it decreasing? What's happening to the state of our world? And they want to know that for their whole country. And we've never measured that for a whole country before. Usually conservation is efforts on particular at-risk species. So for a country to be like, we know the state of our biodiversity, that's actually really, really hard to do. And you need models to do it. You cannot answer that question without models. And I think SDMs play a huge role in all of this. One of the reasons that these SDMs tend to work well for this is because they're aggregate metrics, and aggregate metrics, if there isn't bias, that's a separate thing, which I'll talk about later, they tend to be really stable. So you don't actually know, this is an example of a paper that was published last year where they did an assessment. So this is at-risk species, but they basically said, what's the richness of at-risk species around the world? And then they took an IUCN assessment from 10 years ago, redid it, turns out patterns exactly the same. We know where the biodiversity hotspots are in the world globally. We don't know it if you get really fine scale, but we know it kind of overall. And so that's why it's nice, because we can make a complete map with our models, give it to a policymaker, and be reasonably confident that that result's not going to change that much. And I think weighted endemism is particularly important because it's especially stable. Does everyone know what weighted endemism is? So weighted endemism is a super simple and intuitive metric that I love it because it's intuitive and you can usually explain it to people. I'll give you the harder version, but basically it tells you how much unique biodiversity that you have in a grid cell. It can be for one species, it can be for all at-risk species, it can be for all species. And this is the way it does it. So if these are all your grid cells, and here's our tree frog here, and our tree frog is only found in one of those grid cells. It's a highly endemic species. So people use the term endemic, like, are they endemic to an area? Weighted endemism is like a scaled version of that we can use with SDMs. And so in this case, you would have a 1 here because the species is entirely contained within this grid cell. So that grid cell is obviously really important to that species. And then you can kind of sum it up. So you know globally how much of that species distribution is available and how much is protected. So let's move to our another sort of more common frog here. And this one, in this case, it's taking up 3 of those grid cells. So you divide it all up. Super easy metric, right? And so in this case, you're like, for conservation purposes, you're like, well, we don't really know which to go to. Any of these are kind of equally as good as the other. But if you would pair it with that other species, then you kind of know exactly that you care about this species is like, it's too hard. We can't do anything about this efficiently. This species we care about because we know exactly where to go to protect it. So it's a really efficient metric, and it scales really nicely, and it underlies basically all spatial planning algorithms. They worked differently, but underlying that idea is the idea of weighted endemism. So that's really, so if you're making your SDMs, this is definitely something that you should calculate. We talked about this evaluation. So calculate species richness, then calculate endemism and see what the differences are. And so why do it? So because it's based on a stable metric, because the time is right. We need to do this now because countries are trying to get to 30 by 30. And I know that EU is a little bit different here, but they're still doing a big effort to get to 30 by 30. And probably never again will that happen. So we've got to be there to help people make these decisions. And then, obviously, whenever we talk about conservation, what to keep in mind is that it's just not always about biodiversity. But at least if we give policymakers numbers, then at least biodiversity can be part of the decision. Is it okay? Sorry.
Yeah, please.
Okay.
We went in the TV to shut it.
Oh, I thought you were like, this slide looks like to me. Get off of it. Okay. Fast forward. So one of the things we can do, we can map biodiversity, we can also map other stuff. And a lot of the techniques that we've been talking about are useful for that as well. This is just to show, so this is in Europe, and what we did for this study, this is one of my PhD students when I was in Grenoble with Wilfred Thuyer, Louise O'Connor, and she decided to do spatial planning for biodiversity based on weighted endemism, and then do spatial planning for ecosystem services, and then put it all together and see if those conservation priorities matched up, and they didn't, not a little bit, not at all. So this is the overlap. This is sort of the species version of it. This is the NCP, cultural NCP, regulating NCP, and this is the overlap of all 3, which is less than 1% of Europe, in the ideal situation. So a little bit depressing, but within this, there are some really interesting places that would be useful for conservation. And so in some ways, having this little narrow slice of information is useful because you're not being like, you could go anywhere. You're like, these are the places that protect both biodiversity and ecosystem services. And Louise also found that if you protect species, you will get ecosystem services from that. If you do the opposite, if you protect ecosystem services, you will not protect biodiversity. And the reason is because biodiversity is much more clustered than ecosystem services, which are much broader layers in general. So sometimes it doesn't match up. This is an example from Canada done by Isaac again. So again, talk to him about this. But this is where we took all the groups of terrestrial vertebrates, butterflies, and plants, and then we did this kind of same exercise of what's protected now, what would you need to protect to get to 30 by 30, and pretty much if you choose birds, like I'm going to protect birds only, I'm going to protect butterflies only, more or less all of them give you the same answer. So it doesn't really matter, even if you're like, I care about at-risk species or I don't. This is what I'm talking about, being kind of a stable solution here. And you always get really big gains. So about 13% of biodiversity is protected in Canada right now. And we could have about 60%, which is a huge gain and completely unrealistic. But even if we were to get some way there with 30 by 30, it would be a huge gain. So we know that it doesn't really matter the biodiversity part, which is kind of dissatisfying being a biologist because you want to think that some species matter more, and it kind of doesn't, at least at this scale. But if you instead decide, I want to make this optimization and I want to do it region by region, either province by province or ecoregion by ecoregion, you get much, much less potential gain. And so this is basically like saying we have to kind of work together, which is hard to do, obviously, but as a country, I feel like this is the time to do it because a lot of countries are trying this kind of national planning, which they've never tried before. So this is like a huge difference, but still, even if you get kind of half of this, you can be doing very well for biodiversity. Indicators, I'm not going to talk much about other than to say there are lots and lots and lots of indicators. Most of them do more or less the same thing, and almost all of them are based on the same data. And very few of the ones that are used in any of this policy have any biodiversity data whatsoever, like zero. So most of the indicators that are used are either things that, in the case of industry, they're things that they can tick off of a list, be like, oh, we did 3 green projects or something like that. Those are the kind of indicators they use. For the more biodiversity ones for GBF, most of them are land cover based. So how much, developed area do we have? How much roads, things like that. There are some that have been developed and proposed for biodiversity. So Living Planet Index is one example. But almost none of them, Living Planet Index didn't quite make it into the framework. Almost none of them that use biological data are in anywhere. And it's really hard. We've tried to do this with Canada. It's hard to convince governments that they need to have biodiversity data and biodiversity indicators. It's a bit counterintuitive. But I want to just highlight these. So these are the targets. And I just highlighted here, this is target 3, 30 by 30, what we've been talking about. But pretty much all of these, we need to understand degraded ecosystems, loss of area of high biodiversity importance. Almost all of those can use species distribution models, and some kind of have. And so they've said these are the ways that we want to reduce threats, and these are our targets for doing so. To me, SDMs work in so many more of those than for what the indicators have been proposed for. And this is another, I just want to point out, this is a really, really important target. This is how much data do we have to answer the problem? So the data information index, which has been derived a few different ways, but it's really important, again, for all the things we've been talking about, is getting more data in the right areas. And that's just kind of to show how they've thought about getting this. So the species status index is one of those, and they measure that with range maps. The other thing that I'm really excited about right now, mostly because we just have a paper that we've been writing and it's accepted and coming out, I believe, next week.
Yeah, next week.
Woo, very exciting for a long process. It was a long one. Yeah, it was very political too. Yeah, so I really think that so this kind of shows sort of these are different ecological processes that you would expect to be important for extinction risk, basically, more or less. And so you have kind of behavior shift So this is something like, a road goes in and you have movement behavior is interrupted. And then you'd have things like demographic shift where it's working its way through the population. And then eventually you have entire population declines until the point where you have a local extinction, then a distribution change if you have a lot of local extinctions, and then all the way to the actual extinction of the species. And so most of our indicators right now are kind of over here. So the red list index is a really good example of an indicator that's used all the time. And it's a great indicator. It's about as good as we can do, but it relies on reassessments, which happen like every 10 years. So it's a very, very slow lagging indicator compared to some of these others. And then these are kind of more SHI, which is driven entirely by distribution. And then there's some abundance things. And then there's the genetic diversity indicator. And so the point is, like, we need to get more information in here. And I really think we need to get more models, because we're never going to have the data to measure these things directly. But people are really distrustful of models. And so we just need to convince people that we need models because they represent the ecological process and they can help us understand and predict through time. And so I think SDMs kind of fit there. And I really think that movement data and SDMs work together so well to do this, because movement data, we're getting more and more of it. It's representing all of these different processes, like early warning behaviors, down to habitat use and dispersal. And I'll just really quickly talk about a project. So this is one of my master's students, Samara, and she was trying to kind of make these models, and it was an experiment to see, like, can we make a model-based indicator that would kind of work to tell us what's happening with movement globally. And so it takes like individual animal movement tracks to create a home range estimate, and then you can kind of imagine multiple home ranges. And the trick is to kind of take this data and make it at a species level, and then you can say, well, if we understand that this individual contracts its movement or something based on land cover change, well, we know what land cover change is doing. That's easier to model, and then you can kind of estimate those things. And so for globally, we've known for a long time, overall, depending on the species, but overall, if you have more human development, it does shrink home ranges, with the exception if it gets really small, then an animal actually moves more. But overall, it does contract ranges in the end. And so then you can kind of map what change in home range would you expect for a change in HFI in this case. And then you can map out HFI in time and be like, well, what would we expect that to do? And then you scale it to the entire species distribution. And it kind of works. And of course, it's depressing. Yeah, but it's depressing. But yeah, so you have the mean HFI through years. And for this, in this case, and you can kind of then map it out in space. And the nice thing about doing that is the indicator that we use now at that kind of scale is the species protection index. Usually it's based on the IUCN extent. So what they do is they take the IUCN range extent and they sort of filter it by protected areas. And then they calculate how much they have a target for that species, calculate whether or not that species has enough of its distribution in protection. It's a very, very blunt metric. And if we use SDMs, we can kind of refine that a little bit more. And then if we use estimates like this, where we have the SDM, so we kind of know where the habitat is, we know where the movement and the sort of survival area is expected. And we could, in theory, I say this because we haven't done it yet, we could improve the species protection index, I think. This is, if you take those results and you say the proportion of optimal home range that's been lost, and this is an aggregate metric for all those species we have data for, mostly large mammals, globally, and you can kind of see that a huge portion of the kind of movable area for species has been lost, especially in areas that have obviously a high human impact. Okay. So that's like a very glimpse of indicators. But again, just, when you're making these models, think about what are the outputs. And I think one of the nice things about what you're doing in the machine learning community and just in general is like you're so good at these big data and visualization things. And it very rarely do we have that information in these talks. Usually it's like one person speaking with like an IUCN range on the slide, and that's great. And that's what they're talking about. But it would be really nice to have like what is the information that we could give to these people that are making decisions that is really easy to visualize? And then I can't talk about anything really right now because I'm in Canada and our data sucks, honestly. It's terrible. It's worse than you could possibly imagine, and I can give you all kinds of statistics to back that up. Like 93% of Canada doesn't have a single mammal record. Not at all. Anyway. But I think it also really applies kind of everywhere. I don't really know. I've never heard a talk where they're like, we have enough data. We're good. We're good here. Like that just never happens, right? And so we also know we don't have a lot of people to do this. How do we collect data where we really need it for the question that we're trying to answer? And so that's what we've been thinking about a lot over the last few years. And so there's various versions of adaptive sampling. And the reason I want to talk about it here is because I think there's a lot of potential in the methods that you're using in reinforcement learning or other things where it's like you're actually, that's what the model's trying to do, is kind of adapt to the situation given targets. And I think it's barely used. It's honestly not really used that much in the ecological literature. It was used way more in like the 80s when they developed these kind of ideas about that we need to optimize our monitoring. And it's just, it's there, but I think it's like too practical of a question maybe to be interesting enough. I'm not sure why it is, but we need to do it. And this is just to convince, I was just pulling this plot up earlier, just to convince you this is Europe. And these are the FLOTS data that we've been talking about. So this is the EVA FLOTS data with, millions of points, just the best plant data set in the world anywhere. And this is just what happens this is that 1 kilometer. If you build distribution models on the plot data, and it does change depending on what kinds you make and how much you bias correct, but it doesn't change that much. And this is the kind of richness pattern that you get. So the Alps are obviously really important in the plot data set, and kind of, the Pyrenees and other mountains. And then if you look at all of GBIF, this is the richness pattern you get. Same method, right? Same method of building your distribution models. Obviously, there's a lot of stuff that's inflated here that we can see. But one of the reasons that this is more is because botanists love the mountains. There's so much bias in these two data sets that it's hard to compare this. And this is in Europe. And so, I'm not saying there aren't good ways to put this together, because there are, I think, but it's just really stark to me that they're so different. And so I think even here, we need to think about sampling, and we need to think about adaptive sampling. Am I okay? No one's paying attention. They don't care. Okay.
Sorry, OCR means something very different.
Now I'm curious.
It's like when you're recognizing text in images. That's like,
Great.
OCR stands for optical character recognition. Yeah.
To us, OCR is occur, which is occurrence checklist range validation. Noah worked very hard on that too. It's pretty cute. So one of the reasons that we talked about before is we can't just use GBIF data and cut it up and validate on the other part of the GBIF data, because it's all biased in the exact same way, because a lot of it's citizen science data and other reasons, other data sets are biased too. So this is kind of what we've proposed as a not a perfect solution, but for Canada, and I think probably a lot of other places too could use this. And so what we've done is you have your GBIF occurrence data. That's kind of the standard validation that we would do. And then this is really the key, is this park occurrence data set. So this is a data set that I think we have in Canada, and I think we have a lot of places in the world. So if you go to your local national park and you ask the biologist there, and they can almost always tell you what species are there, way better than a model that we could make for them kind of top down. And this level to me is really, really important because, first of all, it's a meaningful unit for conservation, but also it's much more at the scale we kind of need to match the models that we're building, at least for the data that we have. I'm not saying in Alexi's model, your models are much more fine scale than that, but in a lot of places, like the biodiversity data, especially if you go back more than 10 years, is not that locally precise anyway. And so I think this is a nice kind of intermediate scale where we have true absences, sort of. To me, it's the most true absence you can get at this big scale are these part checklists. And it's not like I don't love that because there are problems with this as well, but I really think that we should try to get this together to do this for other places too. And then of course there's just the IUCN range or whatever expert range outline that you can use. And so what we did here is we modeled the SDMs for the terrestrial vertebrates of Canada. We also did climate change projections. That's a whole other thing that we can talk about later if we want. And then we kind of validated them for the 3 different occur validations. And the other nice thing I'll say about these kind of checklists basically is they can come from anywhere. So the key biodiversity areas is global. They also have checklists for all of the key biodiversity areas. That's one really good example of where we could look to find this information. And there's a lot of traditional knowledge and local knowledge of a species list. It's one of the easiest things you can ask for someone. They know immediately what you want from them. You're not asking them some obscure question about trying to use expert elicitation. And so I think it's really an invaluable but kind of challenging to model because you've got a polygon, basically. So you have a polygon with true absence. What do you do with that when you have a point-based, raster-based system? And so one of my PhD students, Noah Whiteman, is working on this, and he's come up with this kind of scaling approach, which it basically takes the outline of the area where we know the true negative information, and then it kind of, depending on the raster, there's a clustering coefficient, which explains a lot of this actually. So there's a kind of clustering coefficient that will kind of link up basically the raster of the predicted probabilities with the true absences. And this is very much a work in progress, but he's got some really encouraging results that show kind of, what we would expect to see in those 2 things and allows those 2 scales to be comparable. And this is just to show you the results of this. And so this is your kind of, the validation score, whichever one it is. And this is an SDM that's not really corrected. It's got a tiny bit of correction in it. This is basically like a maxent model if you would just run it out of the box. This is target background correction, which all of you are familiar with. This one is the covariate correction. This is something that is not recommended that you use. The only time you would use this is in really extreme situations of bias, which we have. So what this does is you actually stick the bias variable directly in the model, run it like it's a covariate, and then you change it to predict forward. So an example of this is distance to roads. So if you put distance to road into your model, and then when you make the projections, you say distance to road equals 0. It's something that, you're totally in extrapolation. You never want to do this except in these extreme situations. The only other time I've used this is in Australia, where we had also bad data. So if you use the maxent model and you validate to GBIF data, it's going to say that the maxent model is really great. The AUC here is very, very high. And so, yeah, overall, of course, if you validate to biased data, you're going to get a biased answer. And then this one, actually, the bias covariate, the crazy one that I was like, oh, we should try this because it seemed to work in Australia, actually validate a lot better to both the checklist validation and the range validation. And the reason is because it takes the species, the northern distribution of the species, the northern projected distribution of the species, and ramps it up way further north than you would given the data, but it actually gets it much more accurately than not doing it. And so again, it's not necessarily a recommended approach, but I think having these kind of 3 things just helps a little bit you understand the problem. And hopefully at some point we can do all of this with the point data itself, but it's nice to have separate validations. The very last thing is I can't not talk about this. I'm going to talk about it more next week in detail of the talk, but we have been trying to organize citizen science mostly scientists actually, to go out and collect data. And so we started Blitz the Gap last year. We did one summer of sampling last year. We had a whole bunch of challenges. There were a bunch of people involved in this. But one thing that's really nice when we actually analyzed the data, it took us like 6 months to get the data. Once we analyzed it, it actually did kind of work, which was great because we just thought it'd be fun and to get people engaged, but it actually works. So this is the Observations that were collected in Canada last year from iNaturalist. So this is all through iNaturalist. And then this is kind of the different numbers of observations. And then these are the people, so the Blitz the Gap effect are the people that signed up for the main project of Blitz the Gap. And there are about 800 that we considered to be one of the projects. And you get a little bit more efficiency here, but where you really get the efficiency is BioBlitzes. So BioBlitzes, if you take the same person outside of a BioBlitz and you put them into a BioBlitz, no matter if they're a super user or not, they approximately double their observation, which is great. And then the purple here is where we actually paid people to do stuff, which works actually, surprisingly. And actually the biggest effect is not us at all. It's the BC Parks program. They've been paying people for 6 years to go out and sample, and they've done an incredible job. They've gone from they know nothing about northern biodiversity to they have thousands and thousands of species, and they hit 1 million observations last year, which was impressive. So I think this goes to show, though, if we tell people where to go, they will go there. And I think we're still working on how do you make it fun for people, how do you make it into something where people want to go collect. But I think we can do it. I think it shows that it's possible if we are strategic about where we're asking people to do this. What would you do if you wanted people to go out and collect the plant data? They're already going to be out there. They're already botanists. They like to be out there anyway. If they could go to a slightly different place, then they probably would. This is just what I was doing before. So we're trying to figure out in Canada how to then optimize this so we can actually send people to do things specifically rather than just these kind of broad challenges. This is just an example. I've been working with the amphibian data set for British Columbia, which is a nice data set because there's almost no records before 2019. And so this is just this is basically your SDM validation here. I've just left it as a ratio. So this is a predicted suitability. We have 0 absences, right? No, we have, yeah. So this is the predicted suitability. So this is when you go out and sample, this is the predicted suitability of those sample rasters compared to different kinds of backgrounds. This is actually compared to the background of that ecoregion that it's found in. And so, things up here are better, right? And then the trick here, I'm not sure, you can tell me if it makes sense or not. The trick here is that you also measure survey completeness. And so that, depending on how your model validates, you can use that threshold to say, how much area would we need to sample that hasn't yet been surveyed based on that? And so the better your model is, the more that area decreases, right? And the more you're really kind of sure that you're going to find it in a smaller area, which is a more efficient way to go sampling. And so you'd expect kind of through time for it to move in this direction. And so what I did is I just put different time chunks that we have started in. Do I have it here? Yeah. So starting in everything before 2009, which is not much data, honestly, I fit the model on that, and then I would test it on the next year, and then group it all, and then do that again. So this is the 2009 validation, 2014, 2019. So between really, iNaturalist took off a lot in 2020. When people were really bored in the pandemic. And then the 2022 to 2025 is the final validation. And so each of these you have these trajectories for each species, and you can kind of tell, at least that's what I think, without thinking about it that much. I think it's a nice way to kind of see how we're doing for each species. And then, of course, you can summarize that for different groups. So this is the Western toad. The Western toad is the most widespread amphibian in BC. Amphibians don't do very well in BC. It's too cold. And so this guy is really, other than the wood frog, which can literally freeze itself and be fine, it does pretty well. And so what I was expecting to see is this kind of thing. But what happened, of course, is not that. It actually declines. So the first years were actually better. And the reason is because they were for museum specimens and people that would go explore. And then when citizen science starts, it actually makes the models worse for validation because you have a whole bunch of more bias that comes into it. So it actually kind of got worse the first time series. And then it starts kind of going the same direction in terms of survey completeness. So we're sampling a lot more area, but we're not necessarily getting better models. And so my question is how do you do this? And these are different types of models. It's an ensemble of a bias correction. This is a MaxNet model. This is actually an updated model where we use where people have sampled to kind of update the models as you go. as expected, this one's going to do better. But still, none of them are going like in the right direction. And so the question is, how would you do that? Where would you sample to get things going, to improve? And this is for all species or all amphibian species. Again, so here's our western toad is right here. And you can see like overall, it's kind of the same. So most species have a better model than the western toad, which is very widespread. Most of these species are just in the south. And so they have, like, we're starting off, these are pretty high. I didn't put the AUC here, but like, they're pretty high. They're pretty good models overall at this scale. But again, they're kind of like, we're getting more samples, but not really like getting a better model. And this is an interesting one. So this is the bullfrog, and this is actually invasive in BC. And if you go and look at it, like it's really just spreading and spreading and spreading. So it's not surprising that the models did really badly at predicting where this thing was. And so kind of interesting to see, like, I wonder if you would kind of do this for more species, if you would find that signal where invasive species are just really, really hard to predict. And then these are the kind of more rare things here, but some of them, this is the rough skin mute. And so this is, it's really hard to detect, but they've had a couple of really nice survey efforts. And once it gets into iNaturalist, then a lot of people find it. Anyway, so that's just one of the kind of examples of really cool, I think, ways that we could think about and be creative about targeting effort for improving species distribution models. And then you can kind of map it out. So this is the value of information. So this is where you would go for the western toad. If you go like here, you're going to improve the model a bunch, basically.
Are there roads there?
No. Not only that, that's Switzerland. So if you take that and move it here, that's the size of Switzerland. And that has zero sample. There are no samples even close to there, not even a little bit. So it's not an easy problem, but the thing is, this is all pretty homogeneous environment. So like all we need are a handful of points there. We just don't have them yet. And so what I'm trying to work on now is how we get effort into this, which honestly, I have no idea. I've tried a couple of crazy things and they didn't seem to work. So if anyone has any advice, that would be great, or ideas.
Do we want to send people to untouched natural areas?
Yes.
I think with a
Yes.
single person doing
Yes.
a structured survey, totally, that's like.
Yeah.
Most science has been done. And her point is we only have 3 points. You don't need to send it.
It's also like 2 people. There's only 2 people in Canada that would go here.
Yeah.
And we know both of them. Yeah. Anyway, that's all I have. So thanks everyone. And thanks to my lab and all of these wonderful people. And thanks everyone for listening.
Thank you very much, Laura, for your talk. And I'm sure that there are a lot of questions. Yes, please.
So I have 2 maybe like probably very simple ones.
Yeah.
One is especially going back to the validation of the occur OCR model. Is one place, have you thought of validating it on National Ecological Observatory Network data? And so US has, in addition to parks, right, sites where they have very complete lists of everything, including invertebrates, right? So, and for some states, they also have very good natural history survey data. So I wonder if that could be a good just sanity check validator for a lot of these models.
It's a sanity check, but there aren't enough spatially to, like, we need more spatial coverage of validation data sets. So yeah, it's a nice sanity check for that area, but it's hard to validate with it.
It's as good as parks, no?
There are a lot more parks than long-term monitoring. There aren't that many long-term monitoring.
81 parks or long-term LTRs and Neon 81 in US.
Yeah, but the US you can use a lot of.
Right, just for mathematical validation, essentially, for model validation. The other question was about movement. One of the biggest kind of use cases and conversations about movement are about migratory species, migratory and semi-migratory species. But that's also, we quite often are not taking into account the three types of migration into SDMs, and particularly into the indicators, all kinds of indicators. Or rather, the question is, can it be useful? When is it different? Like, as we're looking for, especially as we're looking for indicators that take movement into account, is migration different than local range movement? What does it tell us? How can we use it as an indicator?
Yeah, I mean, it's a good question. So a lot of the ranges that are used are breeding only for birds. So a lot of like the big richness estimates and all of that come from IUCN breeding ranges, right? So that's to start, like that's the baseline. And there are different ways. I mean, you can very simply include migration in like summer range, breeding range, and there's a lot of work kind of on that. But yeah, of course it matters, right? And we have pretty good estimates of it now. It's hard for an indicator, and this is a whole conversation, but for an indicator, it's like you kind of want something that's robust, and it's hard with migration. It's such an ephemeral, literally, it changes so much that. So one of the things that they're working on, which I think is great, is kind of almost like a corridor. So we have these big migratory pathways that are very well known, and we need to have kind of a red list of those. And I think that's a great start to this question. But movement itself is hard to like translate it to an indicator. Again, I think we could kind of do it through the distributions somehow, but it's tough.
So it is, and I'm seeing a lot of that work actually not in conservation, but related to epidemiology, and like right now, a lot of that work is and joint species distribution modeling and super fine grain, and particularly like the there's different terms, I forget what they are, long migratory versus mesomigratory versus like short migratory, the range very short in the context of avian flu, right? For example, or the vector-borne diseases. Again, there's a lot of that work. That how much of it is translated, or how much cross communication is there?
I mean, I'd say there's probably a fair amount. Like, I mean, we use the same models, right? We use the same models for spread, as disease models, occasionally. And there's, things like One Health that's a lot of what they think about. And I mean, to me, they're kind of further ahead than we are for biodiversity, but maybe not. I'd say in general, they have better data in there. So, there is some crossover.
That was the question. Can we borrow from epidemiology and One Health modeling approaches and data to inform biodiversity?
I think the modeling approaches are pretty similar as far as I can tell, and the difference is
They're
the
a lot
amount
more
of data.
fine-grained.
But I think you can't do more fine-grained models without more fine-grained data.
We'll do an individual-based model if we have the data. We just never
They do, yeah.
really have it.
They have.
They
But
have.
do we have?
But that we can use, like, I don't know if we're looking there, right?
Yeah. Yeah, I mean, I do think the methods are they're exactly the same, actually, for a lot of the spread models are the same. And a lot of people do both, too. Like, I mean, I think you have Nick Golding's coming to mind, but he came from, that, and then he got interested in ecology. I think he's gone back now. It's kind of back on the ecology side. Sorry, I'll let you.
Yeah. So I was wondering, so maybe one way to get more stable indicators would be to reduce the set of species considered. So like to have a subsample of species everybody monitors. And so, yeah, what's your opinion on that and your thoughts?
I think that's a really good idea. Well, we all love all species, and that idea has been around for a long time in indicator species, right? And yes, they're biased. They're a biased set of species, but they're also kind of biased for a reason. And I think we should. It's kind of, it does change the kind of idea of it. But yes, if you can figure out what your indicator species are, which is a question in itself, of course we should make sure those are monitored. You would definitely want to put your money into a few species and
So an agreement problem, I guess, between different.
Well, it's also like which ones to choose, because if you ask, if you have a session where you're trying to figure out what your indicator species are, at the end of the day, you're just going to have like a fight between like the bird people and everyone. It's literally
Actually, so
invertebrates.
yeah. I was just talking to a bunch of the bird people, and even within there, they're like, okay, we're moving away from taking a list of indicator species and trying to come with like an indicator types, functional. Yeah, a little bit more functional, which I mean, if the bird people are doing that because they don't have enough data.
But it becomes even more dispersed because then you have like all the others, it's hard to choose.
Certainly.
But I think we should. I think it's one of those hard decisions that kind of needs to be.
I think I saw another hand before.
Yes, please. I just wondering if you have an idea for also alternative data collection methods that maybe could supplement. Because you said like some areas are just really hard for citizen science.
Mm-hmm. Yeah, I mean, there's a lot of like technology solutions, but they're really hard to deploy in those areas too. So, I mean, there's stuff we can do with satellite imagery, of course, but getting on the ground data is really hard. I think it needs to be funded, honestly. And it's a pretty cheap way to collect data. If you think about how much money goes into these big monitoring programs, like they're hundreds of millions of dollars. And so I think there's a middle ground where we can pay people to do it, to go collect data in these areas that haven't been collected before, but also to maintain a record where we have started one too. You don't want to lose that. And unfortunately, some of the funding is going away for that, which is too bad, but it's just, it's really hard with science funding to maintain that data collection through time.
You have to convince me your sensor is smarter than a person before I'm like, it's worth it. Cost-wise.
Yeah.
What are your thoughts? And I know it's a controversial topic too, where there's an idea now to require businesses, corporation industries that are in the environment to provide data as part of the taxing. And I mean, I can go both ways on this one.
I don't know how I feel about any of this corporate involvement, to be honest. I think it's great that it's a thing now, but they're companies, and it's not even necessarily that they don't want to do the right thing. It's just, and I think part of it's on us, I will say, part of it's on us to not have given them an easy solution. Some of it's like, oh, it's too complicated. We can't say that anymore. We can't be like, oh, it's too complicated. We don't understand. Like, we're talking about it here, but like when you're talking to someone, you'd never want to be like, we don't understand as a scientist, because then like someone is going to come in and model some stuff, and like we've all probably talked to these people where you're like talking to them and you're like, you're doing what? They're like, oh yeah, I'm modeling biodiversity. And I'm like, how are you doing that? And then they have like the star metric, they have like metrics.
Just providing data on species.
Yeah, but some of these companies have already been hiding solutions they had just to make profit. So why would they give data to show a hotspot where they want to log? They will never do that.
They won't.
Say there is nothing.
Third-party verification.
I would not trust that at all.
There are exceptions. And so the best monitoring we have in Canada by far, by an order of magnitude, is paid for by oil and gas money, and it's in Alberta. And we laugh, but it's great monitoring. And yes, who knows what they do when there's a place they really want to drill? I don't know. But for everything else, they have a grid, they monitor it, they have acoustic report, they have all this stuff out, and it's the best thing we have.
But did someone test the data compared to other areas?
But testing verification is easier than initial collection.
Anyway.
Yeah, you would be careful.
Interesting discussion. Do we have other questions, maybe from students or early career scientists? Maybe the very last one. Yes, please.
Mine is simple, I guess. On the amphibian plot, what was this axis again?
It was Basically AUC.
Say again.
It was the evaluation of the model.
Using
Species.
like the observations that you have, like
Yeah.
How much the model says they're favorable in the places that they're found compared to the places that just other random locations where they are.
Exactly, which is kind of the only thing we can sort of do. So yeah, it's just the ratio.
Yeah. I don't know if this is feasible or not, but could it be that like when you had just the more expert data at the start and then this sort of gap went down when you got more data, that maybe the initial observations were more like focused in a region where people thought they would find this thing, which sort of artificially created like a really tight little area that you'd find it. And then as you got more observations, it just naturally got more diffuse. So would you like potentially expect it to go down as you got more data, or?
It's a really good question. Maybe.
Yeah, I don't know.
Maybe it would. I mean, it's so like the observation process is so biased, and that bias changes so strongly in the different year time frames. So like, yeah, maybe, you know, and at some point it's like it's on us to do the bias correction, right? Because if you collect more data, that should be better, like kind of always, right?
Yeah.
I mean, I know it's not really the way, but like we should want more data.
Yeah, absolutely. Like ideally, you'd be able to make use of all the data you have, and you don't want to have to cut some of it out because you've got too much in one area or whatever. So
and the data where it's biased, like that's essential for understanding change through time. And so we want to keep collecting that, even though it's messing with our SDMs. Like it's still important to have. But yeah, I don't know. I don't have any kind of null model in here or like default. But I think it'd be interesting to kind of simulate different process-based like collections and see how those trajectories would change. Yeah.
Cool. Thank you.
All right. Thank you.
Let's thank Laura again.